Good day, all of you. In our last class, we saw about ovule structure and types and different structures concerned with the ovules. Today, we will see the megasporogenesis and development of female gametophyte in our class. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mehboob Telangana State. So, in our last class, we saw what are ovules, where are they present, how these ovules develop inside the ovary, different stages in ovular development, and types of ovules in angiosperms, and the structures which are concerned with the ovules, the extra ovular growth such as aril, caruncle, and endothelium, obturator, etc. Today we will see megasporogenesis. So, in our last class, while discussing the development of this ovule, we said that the nucellus develops as a small protuberance from the from the outer layer of the placental wall. Okay, so this is the protuberance, small protuberance. You please follow the cursor. So this is the small protuberance called nucellus. Okay, so one of the cells in the nucellus gets differentiated and acts as archisporial cell. So this archisporium is differentiated from all other cells in its size, and it has a dense cytoplasm and a conspicuous nucleus. This archisporium further develops into female gametophyte, that is embryo sac. How it occurs, we will see now. So while discussing yesterday's class, we said that uh, the ovules are of two types based on the parietal tissue, that is nucellus. Okay, if nucellus is more, we call it as crassinucellate, and if nucellus is very less, then we termed it as tenunucellate ovules. So in crassinucellate ovules, the archisporial cell undergoes periclinal divisions. What are periclinal divisions? Earlier also we discussed what are periclinal divisions? The divisions which occur parallel to the adjacent cells are known as periclinal division. So, in crassinucellate ovules, where the nucellus is massive, okay, the archisporial cell undergoes periclinal division and forms an outer primary parietal cell and an inner primary sporogenous cell, same as the case during microsporogenesis. Here also, the archisporial cell undergoes a periclinal division, resulting in the formation of an outer primary parietal cell and an inner primary sporogenous cell. But in tenunucellate ovules, where the parietal tissue is very less, the archisporial cell directly acts as megaspore mother cell without any further division. Okay. So in crassinucellate ovules, here the primary sporogenous cell acts as megaspore mother cell, and the primary parietal cell undergoes many periclinal as well as anticlinal divisions, resulting in the formation of a multiseriate parietal tissue. Okay, so this is how the archisporial cell develops into a megaspore mother cell. The megaspore mother cell, it is in diploid stage, not yet meiotic division occurred. Now, the most important meiotic division occurs in the megaspore mother cell. This megaspore mother cell is large in size when compared to all other cells. Then it has a dense cytoplasm and a conspicuous nucleus. Okay. So you can see here, this is the primary archisporial cell. It undergoes a division resulting in the formation of an outer primary parietal cell, and this is the inner primary sporogenous cell. The primary parietal cell undergoes anticlinal and periclinal divisions resulting in the formation of the parietal layer, and this is the sporogenous mother cell. The primary sporogenous cell, it directly forms into megaspore mother cell. This megaspore mother cell undergoes meiotic division. As a result of first meiotic division, transferally two cells are formed. Then afterwards, these two cell stage is known as dyad. Okay, each dyad has two nuclei. Okay, in the first division, it forms a dyad, and in the very next division, that is the second meiotic division, it undergoes one more nuclear division, resulting in the formation of four nuclei. Okay, so these nuclei arranged in a linear fashion. So this is known as a linear megaspore tetrad, as a result of which the linear megaspore tetrad is formed due to meiosis. Okay, meiosis results in the formation of linear megaspore tetrad. Now, usually three out of the four megaspores. What is the condition of the megaspores here? They are haploid in nature. They are formed as a result of meiosis. So the number of chromosomes get reduced to half. So they are haploid in nature. All the four megaspores which are formed are haploid in nature. Out of four, usually three of them get degenerate and only one remains functional. And this functional megaspore undergoes further development, resulting in the formation of a embryo sac, that is female gametophyte. Okay, so usually uh, the megaspores which are towards the uh, micropylar side, they get degenerate, and the one which is towards the calygeal side becomes functional. Okay, and in some of the plants, for example, the plants belonging to Onograceae, the microspores which are present towards the calyzal end gets degenerated, and the one which is present at the micropylar end becomes functional. And based on this functional nuclei, different plants show different types of developments. For example, in Rosa, the second megaspore remains functional. That second megaspore, which lies towards the micropylar side, becomes functional. In the same way, in Gloriosa and Casuarina, either all the megaspores or only one megaspore becomes functional. Okay, and in Aristolochia, the third megaspore from the micropylar end becomes functional. Whatever may be the number of the cell which is functional, but the further development is similar in all the uh, species, all the types. Okay, so now this functional megaspore develops into female gametophyte, that is embryo sac. What happens here? Um, Again, here on the basis of the number of megaspores which take part in the formation of the embryo sac in angiosperms, there are three types of embryo sac development, that is monosporic type, bisporic type, and tetrasporic type. In monosporic type, only one megaspore remain functional and the remaining three gets degenerated. Okay, that is monosporic type. That means only one megaspore becomes functional. And in bisporic type, a single dyad, that means we already discussed that during the meiosis. Uh, in the first division, uh, the megaspore mother cell divides and forms a dyad. That is a dyad. A dyad means a cell with two nuclei, which later on forms two cells. Okay, so uh, here a dyad develops into a embryo sac, that is bisporic type of embryo sac development. Okay, then the last one is tetrasporic type, where all the four megaspores which are formed as a result of meiosis participate in embryo sac development. Now We will see one by one how the further development takes place. Today, first we will see the monosporic type and bisporic type in today's class. So on the basis of the nuclei in embryo sac, uh, there are two types of monosporic type are recognized by Maheshwari, the famous embryologist belonging to India. Okay, the one is 8 nucleate type and the other is 4 nucleate type. What is 8 nucleate and what is 4 nucleate type? In a maturing embryo, if the nucleus are 18 number, then we call it as 8 nucleate type. And in the embryo, the mature embryo, which is formed as a result of this development of embryo sac, only 4 nuclei are present, then it is known as 4 nucleate type of monosporic type of embryo sac. And yes, here in the 8 nucleate type, this type is known as a very common type because it is found in most of the angiosperms. So it is known as normal type. It was first discovered in Polygonum divaricatum by Strasburger. So it is also known as Polygonum type. Okay. So what happened here? The megaspore mother cell underwent meiosis. As a result, 4 megaspores are formed, which are haploid in nature. Out of the 4, the megaspore toward the callosal side becomes functional, and the 3 which are towards the micropylar end, they get degenerated. The callosal side megaspore becomes functional. Okay, then it consists of, you can see here, it has a dense cytoplasm as well as a conspicuous nucleus. Okay, this nucleus lies at the center. This nucleus undergoes 3 free nuclear divisions. 3 free nuclear. What are free nuclear divisions? These the nucleus alone undergoes division, and it is not further carried out as a that means only karyogenesis occurs and no cytokinesis occurs. Immediate wall formation is not observed here. Only the nucleus undergoes division. Okay, these are free nuclear divisions. Only nucleus alone undergoes division, and no wall formation or cell formation is observed. So here it undergoes first mitotic. All these are mitotic divisions. It underwent meiotic division only when during the meiosis that occurs in the megaspore mother cell, and all other divisions are mitotic divisions. So it undergoes first mitotic division resulting in the formation of 2 nuclei. See, this is the functional megaspore where the nuclei underwent the mitotic division resulting in the formation of 2 nuclei. These 2 nuclei again undergo one more mitotic division resulting in the formation of 2 nuclei at each pole. Here polar differentiation takes place. Each nuclei moves towards the poles. Here one is the micropylar end and the other is the callosal end. See, this is the micropylar end and this is the callosal end. So it moves towards both the poles, and there it undergoes second mitotic division resulting in the formation of 2 nuclei at each pole. These 2 nuclei further undergoes one more mitotic division resulting in the formation of 4 nuclei at the micropylar end as well as 4 nuclei at the callosal end. So later on what happens here, cell wall formation takes place. So out of the 4 nuclei, 3 nuclei develops the plasma membrane forming an egg apparatus. Initially, they are called as quartets. This is the micropylar quartet, and this is the callosal quartet, where 4 nuclei are seen. But later on, the plasma membrane formation takes place. So, after the formation of the plasma membrane, these nuclei, which are towards the micropylar end, they form a uh, triplet. That is, 3 cells. 3 cells develop a plasma membrane resulting in the formation of egg apparatus, where we can see 2 cells present on either side of the central cell. Central cell is bigger in size, and the 2 cells adjacent to the central cell are they are smaller in size when compared to the central cell, and they are called as synergies. These two cells are called synergies, and the central one is known as a egg cell. Okay, this egg cell is nothing but female gamete. Female gamete. See, you can see here. You just follow the cursor. Okay, this is the female gamete, and these are the two adjacent cells. And as a whole, these three cells together call as egg apparatus, and the remaining nuclei which is present here in the micropylar quartet, it moves towards the center and is known as polar nuclei. One nuclei from the upper quartet, that is micropylar quartet, and the other from the callosal quartet move towards the center, resulting in the formation of a polar nuclei. Both these nuclei fuse together to form a diploid secondary nucleus. This is the diploid secondary nucleus, and the three nuclei which are present towards the callosal end, they also develop the plasma membrane, resulting in the formation of three cells towards the callosal end, and these three cells are known as antipodal cells. Antipodals. See how many cells are there as a whole? Three cells at the micropylar end, three cells at the antipodal end, and callosal end. These three cells are known as antipodals. These three cells are egg apparatus, and this is the central big cell with a diploid nucleus. So 3 plus 1 plus 3, total 7 cells and 8 nuclear structure is formed. Okay, so this is the polygonum type, which is most common in angiosperms. Okay, then now we will see the other one, which is four nucleated type. Four nucleate type, this is also monosporic type, as already said, and it is discovered in Inothera lamarckiana. Inothera lamarckiana. Hence, it is also known as Inothera type. This type is the most common feature of the family Onagraceae. In this, the micropylar megaspore is functional and it takes part in the development of embryo sac. Okay, the two nuclei which are formed by the division of the megaspore nucleus remain in the micropylar end without polar differentiation. These two nuclei undergo one mitotic division, form four nuclei. The embryo sac develops from these four nucleate structure. Okay, so three nuclei remains at the micropylar end and form an egg apparatus, same as in the case of polygonum diabaric atom. See, instead of forming 8 nucleus, only these four nuclei are present. Okay, so it undergoes, first of all, the uh, megaspore nuclei, which is functional. It undergoes a mitotic division resulting in the formation of two, and these two undergo one more mitotic division resulting in the formation of four nuclei. No callosal, what is that? Callosal quartet is seen here. Only micropylar quartet is present. Okay, so out of the uh, four nuclei in the micropylar quartet, three develops into egg apparatus, and the remaining one moves towards the center and acts as polar nuclei. Here, what is the difference between the 8 nucleate and 4 nucleate type? In 8 nucleate type, you can see from either side, one nuclei moves towards center and it fuses and forms a secondary nucleus that is diploid in nature. But here in 4 nucleate type, only one nucleus moves from the upper, that is micropylar quartet, and forms the polar nuclei, which is haploid in nature. So in Inothera lamarckiana, you will see only haploid polar nuclei. And this haploid polar nuclei, after fertilization, forms a diploid primary endosperm nucleus, whereas in polygonum divaric atom, it is already diploid in nature. So by fusing with one more male gamete, it turns into a triploid primary endosperm nucleus. So you have to remember the difference here. Okay, so this is the monosporic type of embryo sac development. What happened? Monosporic type of embryo sac development is of two types. One is 8 nucleate type and the other is 4 nucleate type. Okay, so The next topic is bisporic type of embryo sac development. See, you can observe here in monosporic, this is a megaspore mother cell. It undergoes division resulting in the formation of two nuclei, and these two nuclei undergo one more division resulting in the formation of a linear tetrad, megaspore tetrad. Out of four, three degenerate and one remains functional. It undergoes mitosis and uh, this is mitosis 1, 2, and 3, resulting in the formation of quartet at each end. And out of 4, 1 from upper quartet and 1 from the lower quartet moves towards the center, resulting in the formation of a polar nuclei. And later on, here, the development of plasma membrane takes place. And these three cells are known as agapparatus, and these three are antipodals, okay, out of which the central cell in egg apparatus is larger in size when compared to the two adjacent synergids. So one more difference you can observe here. Here the nucleus is uh, towards the upper side and the vacuole at the lower side. But in synergids, the vacuole is at the upper side and the nucleus is at the lower side. This is the difference between the XL and the synergids. Okay, so this is monosporic type. And now bisporic type. What happened here? This is the megaspore mother cell. It undergoes meiosis division, resulting in the formation of during the first meiosis only two cells occur. Okay, this is a dyad. This is called a dyad. So this undergoes one more division resulting in the formation of two dyads. This is one diode and this is another diode. Each diode comprises two nuclei. Okay, so one of the diodes degenerate and the remaining one becomes functional. Okay, so in Ilium type, this is also uh, recognized by Strasburger in Ilium fistulosum. Ilium fistulosum. And it also contains 8 nucleus, just as polygonum type. Okay, so this is the diode, functional diode. In Ilium type, the calisel diode becomes functional and the micropilar diode, it gets degenerated. So this is the functional diode and this is the degenerated diode. So in this diode, it grows in size, the nuclei move towards either poles and there they undergo first mitosis division. During the first mitosis division, two nuclei at the micropilar end and two at the calisal end results. Okay, so this nuclei again undergoes one more mitotic division, resulting in the formation of four nuclei at the top and four nuclei at the bottom. That is at the calisel end and at the micropilar. This is the micropilar end from which the egg apparatus develops, and this is the calisel end. So four nuclei, it is wrongly shown here. It should be four nuclei. Okay, four nuclei at the top and four nuclei at the bottom, out of which two nuclei move towards the center, resulting in the formation of a secondary polar nucleus, and the three nuclei which are present towards the micropilar end, they develop the plasma membrane and develops into a egg apparatus with three cells, the two cells adjacent to the central egg cell. This is the central egg cell and these two are the adjacent synergids. In the same way, the top three nuclei which are present at the calisel end, they form into antipodals as the case in the polygonum. Okay, so nothing is different here, same as that of the polygonum type, but here the dyad, the two nucleated dyad acts as a functional megaspore. Okay, already two nuclei are present, so it undergoes only two mitotic divisions. Here only one nucleus, it undergoes three mitotic divisions. As a result, 8 nucleated structure is formed, but here it's already two nucleated structure, so it undergoes only two mitotic divisions, resulting in the formation of a 8 nucleated embryo sac. Okay, here also it is of two types. One is alisma type, Ilium type, and the other is endymion type. Ilium type and endymion type. In Ilium type, calisel diode becomes functional, whereas in endymion type, micropilar diode becomes functional, and the remaining is same. Okay, so this is about today's class. What we have seen megasporogenesis, how megaspore mother cell undergoes meiosis, and how a megaspore forms or develops into a embryo sac we have seen today. Okay, can we have a, small refresher for 5 minutes? That is, shall we again uh, see what we have learned till now? Okay, so this is the archisporial cell which is formed in the nucellus. Okay, this cell acts as a archisporium, and it undergoes a division, periclinal division, resulting in the formation of a parietal cell and a megaspore mother cell. This parietal cell undergoes many anticlinal and periclinal divisions, resulting in the formation of parietal tissue, and this sporogenous, primary sporogenous cell, it forms into a megaspore mother cell. Until then, it is diploid in nature. It undergoes meiotic division, resulting in the formation of four haploid megaspores, out of which three degenerate and only one becomes functional. Okay, and this functional megaspore develops into an embryo sac. And based on the number of megaspores which take part in the formation of or in the development of embryo sac, it is again of three types, that is monosporic type, bisporic type, and tetrasporic. In monosporic, only one megaspore becomes functional and develops embryo sac. In bisporic, two of the megaspore nuclei, they take part in the development of embryo sac. And in the tetrasporic type, all the four nuclei which are formed as a result of meiosis, they take part in the development of the embryo sac. Okay, so monosporic type, again it is of two types, 8 nucleated type and 4 nucleated type. 8 nucleated type is most common, and it is recognized by Strasburger in polygonum divaricate. So it is also known as polygonum type. And uh, for nucleate type, it is recognized, identified in Enothera lamarckiana of Onagracea family. It is a characteristic feature of Onagracea family, where the developed embryo sac has only four nuclei. Okay, then bisporic type, where the embryo sac develops from a dyad. A dyad is equal to two nuclei. So here also it is of two types. One is Ilium type and another one is Endymion type. If the callosal dyad becomes functional, it is Ilium type, and if the micropylar dyad becomes functional, then it is the Endymion type. So this is today's lesson. In our next class, we will see tetrasporic types of embryo sac development. Thank you.